chromatography, as I understand it, with my very limited biology knowledge, is just like a bunch of like a set of different methods to basically separate different proteins from a mixture by pushing the mixture down like a column. What this really means is, if you see here my very beautiful drawing right here, is you basically have this column right here, okay? This is like a tube, and then inside this column you have these beads, okay? These are like small, you know, uh, spheres, I guess. And then you put your liquid down this column right here. And then in one way or another, it will basically filter your liquid down, and you'll get like the filtered, stuff that you might have from it, okay? Now, to do this methodology, there are a couple of different ways. We can filter proteins with this by their size, by their charge, by their type of binding, and also by their hydrophobicity. So we will be going over how each one of these, in very simple terms, works. First, we'll start with size, as this is the easiest to understand. Now, the way that we can see this is basically, different proteins have different sizes, right? Or different molecules also have different sizes. And so these beads that we have here, they might look like something like this, where, imagine this is the close-up of a bead, and then in the bead itself, there are like these pores. You see, like it has pores like that and all of that, okay? And some small molecules like salt would basically fit into these pores, okay? So let's say here, I'll do this in red. This is salt, and salt basically goes into the pores easily like this. This doesn't mean that they get trapped forever, but it means that basically it gets slowed down. While a bigger molecule, let's say, like this blue molecule, let's say this is protein, wouldn't fit. As you can see, it's too big to even fit inside. So these pores are everywhere then in these beads, and then when the liquid goes through, basically the big proteins, they swim a lot faster, you know, and then they land, for instance, here. But then the small, uh, you know, salts might get stuck a lot faster. So, the salts might go in here and here and here and all of that. And then after that, it gets filtered out and into here, for instance, okay? Since it just took a lot longer. And this is how we can filter molecules by size through the chromatographic, I don't know, like size filtration. Another word for this is SEC. Next, we go to filtering by charge, and we can do this through ion exchange chromatography, okay? So I'll write that down here, or also short for IEC. And how this basically works is the beads that we saw before, like these beads right here, they are positively and negatively charged, okay? So if we were to, draw our container here again, and there are beads, and these are either positively or negatively charged. So if it's positive, it's called anion exchange, and if it's negative, it's called cation exchange. And then we basically have again our liquid, our mixture with our proteins where we want to filter it, and that then goes like it passes through, and the proteins then they get stuck basically on these beads because of the charge, because opposites attract, right? So you can therefore filter proteins which are positively or negatively charged with that by controlling the charge of the beads itself. And basically to then loosen it up, you just increase the salt in the mixture, or the pH. You can also do that through the pH. So for instance, add salt, and then it then falls down, for instance, okay? Next, we will go over basically trying to filter it by binding, and this is actually the most exact one. This is called affinity chromatography. And how this works is how I understood it is that like you put in these, you put basically on these beads, something called a ligand, I don't know how to say it. Exactly. So all these beads have this thing attached called ligand. Let's say this blue thing now is this ligand. And basically, this allows you to really, really very precisely filter exact proteins with this. And that is because only specific proteins attach to very specific ligands. So if we then have our protein come through, it would then stick specifically on this ligand. But let's say this, I don't know, this green protein wouldn't stick and then just fall through, you know, because it's not the ligands that it basically sticks to. Okay. Lastly, we can also do chromatography through hydrophobicity. So how this then works basically is that there are some proteins which are more hydrophobic than other proteins. So basically, you put like you have these like greasy beads, and some would then stick very closely to the beads than others. So very similar to this, you would then have the beads with like different types of greasiness, and you actually increase the greasiness of these beads by changing the salt value. So the more salty basically the beads are, sorry, not the beads, but the water is, the more hydrophobic, or the stronger the hydrophobic sticking really becomes to those beads. So the same way would be like this, and then some proteins might stick and some other proteins wouldn't stick. And yeah, that has basically been a very quick summary of different types of chromatography and how they roughly work. Yeah. Lastly, I do also want to say that in practice, you can also combine these different types of chromatography, in a way that might make sense for your experiment. And thank you very much.